1284, 1285. Ah, fresh meat. Salutations and welcome to the fight. What's my name? That's not important. If you're watching this now, that means they called your number, slapped you on the bottom, and sent you off to the war front. That makes it my job to get you ready for the functional amyloids and the nasty bugs that use them. Now lock and load, maggot. It's time to brawl. Now this might seem like the introduction, but we've been fighting this war longer than we can remember. What's crazier is, we've only known the identity of our adversary roughly since the 19th century. We've always felt their presence in some way or another. A cough that's left us breathless, that extended stay on the porcelain throne from that slice of pizza that's been in the back of the fridge for 2 weeks. But it wasn't until Robert Koch, and yes, that is how you pronounce his name, characterized the inimical nature of various microbes that we began to postulate the notion that bacteria can hurt us. Koch's discoveries were further bolstered by the findings of his associate and later rival Louis Pasteur, whose investigation into microbe generation and the pasteurization process to eliminate contamination solidified the foundations for the germ theory of disease. Thanks to the works of Koch and Pasteur, we now knew that some bacteria could cause illness. We also knew that introduction to weakened strains of these pathogens could work to prevent illness as well. But what do we do to treat a person who's already sick? To answer that, we must acknowledge the contributions of Alexander Fleming and Mrs. Mary Hunt. Fleming's accidental discovery, as well as Hunt's sourcing and culture refinement, led to the production of the world's first antibiotic, penicillin. The discovery and mass distribution of penicillin marked the beginning of the golden age of antibiotics, during which time over 20 new antimicrobial classes were established over the course of 2 decades. This ushered in a new age of prosperity that significantly reduced the threat of bacterial infections and increased the average human lifespan by almost 30 years. The best part is, because microbes never adapt to adverse conditions, we could all rest assured that the antibiotic revolution definitively marked the end of bacterial infections as we knew it. Uh-oh. Of course bacteria can adapt! Hell, there were signs of penicillin resistance in Staph aureus back in 1942. For a while, we were able to keep antibiotic resistance at bay by making semi-synthetic derivatives of the drugs we use to treat these pathogens. But now, thanks to medical misuse and wanton agricultural application, these semi-synthetic derivatives are starting to fail as well. If we don't find more effective therapies, the World Health Organization estimates that upwards of 10 million people could die annually from antibiotic-resistant infections by the year 2050. Long story short, the enemy is gaining ground and we are running out of options. The increasing ineffectiveness of current microbial therapies is causing researchers to explore other facets of bacterial pathogenesis. What I mean is that instead of just disrupting essential processes that these organisms need to survive, scientists are now looking to disarm pathogenic defenses. One such defense is the application of fibrous aggregates known as amyloids. Amyloids are proteinaceous complexes that occur across all domains of life. They are insoluble, resistant to degradation, and have the capacity to induce fatal consequences. Yet some pathogens have developed mechanisms using functional bacterial amyloids to enhance their microbial resistance and overall virulence. This revelation has inspired amyloidal pathology investigations for more than 30 years across innumerable taxa. While this work has produced a great deal of useful information for us, there is a lot more ground to cover before we're out of the woods. This presentation aims to review amyloidal discovery, their characterization, their regulation as a means of bacterial pathogenicity, as well as scope the relevancy of their research in reference to public health interests. A physician by the name of Nicholas Fontanus was the first to observe amyloids in 1639 while performing an autopsy. The deceased was a young man whose abdomen swirled with excess fluid and whose skin turned a deep yellow of severe jaundice. During this procedure, he recorded the presence of an abscess in the liver and white waxy stones in the spleen. However, the term amyloid wasn't used until the German botanist Matthias Schleiden coined it in 1839 in an attempt to describe his findings. Schleiden interpreted an iodine sulfuric acid staining reaction in plant cells as a sign that these residues were starchy. As such, he named these deposits amyloids from the Latin word amylum, meaning starch. Unfortunately, this was proven inaccurate 20 years later when the chemist August Kekulé showed that these were molecularly distinct from known starches and contained significant concentrations of nitrogen. Despite being a misnomer, the name stuck. Afterward, wait, hold up! Plant staining? Waxy liver stones? What does this have to do with bacterial pathogens? That's a great question, the answer to which lies in a relatively simple phrase: Congo red. Congo red is a diazo dye used in histological staining assays. Though initially developed for textiles, a German chemist by the name of Hermann Benhold discovered that Congo red dye uniquely binds to amyloids in 1922. When viewed through a polarized light microscope, Congo red stained amyloids appear as red to green bifringent material. This feature is conserved across all organisms, making Congo red useful for detecting amyloid structures in beings such as mammals, plants, and bacteria. In 1989, E. coli curly fibers were identified as the first amyloidal structure in bacteria. Not surprisingly, these structures were later discovered in other members of the Enterobacteriaceae family. By the end of the 20th century, technological advances had made it possible to elucidate further details in amyloid composition and structure. Right about now, you're probably saying something like, okay, now that this clown took a century to explain how we found him, is he ever going to tell us how amyloids form? Well, my caustic and impatient friend, simply put, amyloids are fibrils that originate from misshapen proteins. The likelihood of a peptide to form an amyloid is species dependent. However, over 50 protein residues have already been identified as amyloid capable. The first phase, known as primary nucleation, describes the process by which amyloids transition from a still soluble state to a condensed insoluble nuclei. Elongation occurs when the fibril begins to grow by converting surrounding soluble proteins into insoluble aggregates in a process known as templating. This is often accompanied by seeding, or inducing larger aggregates from smaller nuclei. It is also extremely common for a portion of the fibril to break away from the major complex and stimulate the formation of another fiber in a phenomenon known as secondary nucleation. As the fibers mature, they arrange themselves into a series of parallel or anti-parallel beta sheets that precipitate the signature amyloid cross-beta conformation. The cross-beta conformation creates an interstitial space between sheets that is occupied by an array of amino acid side chains. Amyloid strength and durability is largely attributed to the steric zipper formed by these side chains, as well as the massive amounts of hydrogen bonds that connect them. Additional forces like pi bonds and ionic interactions also help to fortify amyloidal conformation. However, the crux of their strength is credited to hydrogen bonding. Since nature can never seem to follow its own rules, there have been abnormal conformations and arrangements documented for these suckers. Such include the beta-solenoid fibril versus the parallel-antiparallel beta sheets, as well as the low-complexity aromatic-rich kink segments or larks instead of steric zippers. Oftentimes, these variants and their associated structures exhibit atypical behavior like enhanced susceptibility to degradation or heat. Regardless, cross-beta sheets sterically zipped by amino acid side chains are generally viewed as the most characteristic amyloid features. Despite their flawed origins, several microbes have functionally adapted amyloid features to boost their overall fitness. This can include more benign functions like the acquisition of environmental nutrients, as is the case with Streptomyces silicolor. Unfortunately, more sinister bacteria have discovered ways that allow them to thrive at the expense of the host. Take for example Listeria monocytogenes. Listeria monocytogenes is a facultative intracellular pathogen. Upon invasion and subsequent contamination by the host phagolysosome, Listeria monocytogenes will evade degradation by using the amyloidal virulence factor listeriolysin, LLO, to induce pores in the vacuole. This causes phagolysosome lysis, and from there Listeria monocytogenes is free to replicate within the host cytoplasm and continue infecting other cells. Another example would be the pili in Mycobacterium tuberculosis, or NTB. Like Listeria monocytogenes, NTB is a facultative intercellular pathogen. Unlike Listeria monocytogenes, however, MTB doesn't employ amyloidal functionality to invade the host immune processes. Instead, it uses its pili to bind to laminin in the host tissues, which induces pro-inflammatory immune responses. Prolonged inflammatory immune responses can damage host cells, and damaged cells are especially advantageous for MTB infection. So despite not having a direct pathogenic mechanism, the advantageous conditions it creates is why MTB pili are considered agents of virulence. Still, the most famous amyloidal contribution to bacterial pathogens might be biofilms. These are extracellular matrices that are largely comprised of amyloid fibril scaffolds that protect bacterial communities within them from external threats, i.e., the antibiotics we make to get rid of infections. If that doesn't scare you, the list of biofilm-capable bacteria includes E. coli, Salmonella, Staph aureus, Pseudomonas aeruginosa, and even the bioterrorism agent Bacillus anthracis. Freaking anthrax! Other studies have emerged that suggest that amyloids can even be used for things like toxin storage and dissemination. The point is this. This is what we found out so far about amyloid versatility and why they're so dangerous in the hands of morbific bacteria. Don't get it twisted, this partnership doesn't come without risk. At the most basic level, amyloids are malformed proteins that can induce deformity in other peptides. Even if this doesn't increase the number of amyloid precursors, protein performance is critically dependent on proper folding, and disruption to protein conformation could result in inhibited or total loss of protein function in their respective roles. As amyloids mature, the threat only increases. Several studies have shown that amyloids and their associated structures display great affinity for the cell membrane. Unfortunately, amyloid accumulation often results in irreparable damage to the cell membrane. The three most common consequences are illustrated by the ion channel model, the carpet model, and the detergent model. In the ion channel model, aggregates generate nonspecific ion pores across the cell membrane. Research into this model revealed that unregulated inclusion of cations into the cell after passing through these pores could lead to premature cell death. The carpet model suggests that amyloids can create asymmetric pressure on the cell membrane. The result of this is the loss of smaller molecules on the outer wall of the lipid bilayer. The loss of the molecules compromises cell membrane integrity, making it more likely to rupture. Like the carpet model, the detergent model suggests a loss of molecules due to asymmetric pressure created by amyloidal aggregation. However, molecules are lost on both walls of the lipid bilayer, ultimately resulting in cell lysis. Though it doesn't necessarily damage the membrane, it should also be mentioned that amyloids can bind and block receptors on the outer layer of the cell membrane, essentially inactivating crucial signaling pathways the cell needs to survive its environment. Amyloids can even be directly hazardous to nucleic acids. The electrostatic properties of nucleic acids could induce adhesion to amyloid structures that result in single and double helical fractures. This means that DNA and RNA binding to amyloids can cause genetic mutations and/or complete transcription inactivation. It is not an exaggeration to say that amyloidal inclusion means gambling with the integrity of critical structures, essential metabolic pathways, and even replication. Okay, so we know that amyloids can be fatal. We also know that a ton of pathogens employ them functionally without dying. What we don't know yet is how. You might recall earlier when I mentioned that E. coli curly fibers were the first amyloids discovered in bacteria. It turns out that scientists were becoming increasingly acquainted with the Enterobacteriaceae family at the turn of the millennium. As such, it made sense to researchers that additional investigation into this family would provide answers to amyloidogenesis. In 1995, 7 genes were identified as potential regulators for curly polymer production. These curly sequence genes, or CSGs for short, were encoded on two proximal operons in the E. coli central genome, CSG-ABC and CSG-DEFG respectively. Subsequent studies would later discover homologous operons in Salmonella species as well. Seeing that these genes were conserved across Enterobacterales, researchers began their investigations to determine their respective purposes. In their experiments, they saw that disruptions to CSG-D caused drastically lower expression levels for the remainder of the genes in both operons. From this, they deduced that CSG-D was the key transcriptional initiator. CSG-A and CSG-B were found to be disproportionate composers of curly fibrils. While CSG-A was determined to be the major fibril subunit, CSG-B acted as the nucleator that initiated conversion of the fibril from a soluble peptide to an insoluble amyloid. CSG-C acted as a chaperone that maintained the solubility of the CSG-AB complex following secretion into the periplasmic space. Likewise, the CSG-E chaperone performed a similar function as the CSG-AB complex traveled through the CSG transport pore beyond the outer membrane. Lastly, researchers were able to show that CSG-F was the chaperone to CSG-B that allowed for anchorage to the cell. With this information at hand, we had our first regulation template for functional amyloids in disease-causing bacteria. Now that we had an idea of how functional amyloids could be regulated in bacteria, it was time to search for similar systems in other species. This led to the discovery that several members of the Pseudomonas genus regulate these aggregates similarly to E. coli. In a 2010 study, researchers found that 6 genes on an operon in the Pseudomonas genome were responsible for the production of amyloids. In a new and courageous display of creativity, researchers decided to call these regulators the FAT genes, which stands for functional amyloids in Pseudomonas. Like curly, the Pseudomonas fibers are disproportionately composed of 2 subunits, FAT-B and FAT-C, with FAT-B acting as the extracellular nucleator. FAP-F acts as a secretion pore responsible for transporting the soluble FAT-B-C complex. Evidence suggests that FAP-E acts as an accessory protein that facilitates the attachment of the extracellular fibril to the outer membrane. The functions for FAP-A and FAP-D have yet to be definitively defined, but their cytoplasmic locales are indicative of chaperone-like activity, with FAP-A suspected to work identically to CSG-C in curly. If this weren't enough coincidence, FAP and curly proteins both use the same secretory pathway to secrete through the inner membrane. The curly and FAT genes are the most understood regulators for pathogenic bacteria, but they're not the only ones to be identified. A 2012 study determined that phenosoluble modulins, or PSMs, also comprise the amyloid fibers in Staph aureus. Additionally, the inclusion of PSMs is integral to the stability of Staphylococci biofilms. Like curly, the genes for PSM regulation are encoded on 2 proximal operons, alpha PSM 1 through 4 and beta PSM 1 and 2 respectively. Unlike curly, transcription is initiated by the AGR-A gene in the quorum sensing system. Specific PSM functionality isn't cataloged as well as curly or FAT genes. However, we do know that alpha PSM3 is the major subunit in Staphylococci fibrils. Let's shift to more dangerous entities for a moment. Recall that Mycobacterium tuberculosis employs fibrils in antimicrobial resistance and facultative virulence. It turns out that NTB can produce 2 fibril variants, fimbrial low molecular weight proteins, FLP or type 4, and curly-like pili. In reference to the former, the production of FLP pili has been linked to the upregulation of a 7-gene operon. Likewise, the primary component of amyloid fibrils made by Bacillus subtilis, TAS-A, was found to be part of a 3-gene operon along with the anchorage protein TAP-A and the signal peptidase SIPW.
But wait a minute! You said this was all about pathogens! B. subtilis isn't a pathogen!
Well, jive turkey, Maybe it isn't, but its big cousins B. Sirius, L. monocytogenes, and B. anthracis, who also have this operon, definitely are. At any rate, the continued exploration of amyloid regulation across different species is exposing common themes that could prove useful in therapeutic applications. Findings on amyloid structure, gene expression, and their associated pathways have led to novel therapy strategies for combating antimicrobial resistance and bacterial virulence. Let's revisit curly genes, for example. Since bacterial amyloids share key similarities to those of mammals, an investigation was launched to see how well CSGA could resist an experimental Alzheimer's treatment. This treatment uses D-enantiomeric peptides to slow the formation of amyloid plaques in the brain. The study showed that the same peptides could prevent CSGA aggregation by inhibiting fibrillation at the R1 and the R5 repeats. In a separate study, CSGC was found to be structurally and conformationally identical to the transthyretin transporter protein TTR in humans. Knowing that CSGC maintains solubility for curly fibrils, researchers theorized, and later proved, that incubating CSGA with TTR would curtail fibrillogenesis. Now, let's pivot to B. subtilis for a moment. Researchers have so far identified two bioactive compounds that prevent TAS-A fibrils. These compounds, AA-861 and parthenolide, were applied to an anti-amyloid study in B. cereus, E. coli, and B. subtilis respectively. There, they produced similar anti-amyloidal results. Subsequently, biofilm capabilities were abolished for all three organisms. But don't you wish there was a naturally occurring substance that could put the kibosh on amyloid production? Well, my friends, you're in luck, because there happens to be a plant derivative with exactly that functionality. I'm talking about polyphenols. A 2016 study showed that a host of natural compounds could eliminate biofilm capacity in E. coli, Staph aureus, and Pseudomonas aeruginosa by targeting the signature amyloidal compounds of each organism. Of all the compounds, EGCG was the most effective. As a bonus, researchers determined that EGCG had the potential to inactivate the curly operons by inducing unaffiliated sensory pathways on the cell membrane. I haven't even gotten to the best part. Do you want to know what plant we source EGCG from? Do you? That's right! Green tea! That warm cup of hipster self-importance could potentially be a neutron bomb against amyloid-capable pathogens. As you've heard, the options for defense against bacterial amyloids are increasing. But don't let the good news fool you. We still have a long way to go in the way of applying these remedies and understanding these systems. For starters, we still don't completely understand why some polyphenols are so effective against evolutionarily divergent amyloid schemes. While amyloids might share some similarities, the affected peptides in the EGCG study have almost no common amino acid regions. This includes the seemingly homologous curly and fab fibrils. As much as their expression systems might resemble, they're about as related to each other as bat wings are to bird wings. Moreover, investigations are still ongoing to further characterize the less known amyloidal expression systems, such as PSMs and staphylococci. PSMs fall into that variant amyloid category we touched on earlier, in the sense that they are cross-alpha peptides instead of cross-beta. Furthermore, PSMs have an almost paradoxical range of function. In addition to adding structure to biofilms, PSMs can act as surfactants that loosen sections of the structure, allowing for more effective nutrient transport to the cells below. This poses a problem because interfering with PSM composition could induce the undesired effect of fortifying biofilms, making it more difficult for antibiotics to reach the infection. The issues only continue when we transition to Mycobacterium tuberculosis. Debates have emerged concerning whether MTB, curly-like pili, should remain classified as amyloidal. This is because, while MTB curly-like pili play a role in biofilm production and have similar ultrastructure to curly fibrils, they lack the primary sequence homology and typical beta sheet secondary structure. In terms of the type 4 pili, The function of these structures is assumed based on the information gathered in similar complexes for other organisms. Speaking plainly, more studies are needed to categorize what they are and how they aid in MTB pathogenesis. If this wasn't enough, in 2018 researchers found 10 genes that were previously undefined for curly synthesis in E. coli. Through mutant assays, they found that manipulation of these genes resulted in differing levels of CSGA expression. Additional investigations discovered two new chaperones with functional abilities like that of CSGC, CSGH, and CSGI respectively. Keep in mind that the curly fibrils are the most defined amyloid system in bacteria, and we are still making discoveries on it. Besides that, creating treatments against these structures without fully understanding them could result in mutations that make pathogens more resistant. All that to say, we need to make sure that we thoroughly examine their munitions before we step back on the battlefield. But don't despair, we're developing a few new tricks ourselves. In addition to the ongoing studies that I've mentioned, technological advances have given us the means to do things like create phylogenies or predict amyloidogenesis even in novel proteins. These tools and others like them will be invaluable as we prepare for the fight ahead. Congrats, Maggot. Looks like you made it through training. But don't think you're a seasoned vet now just because you can do a couple pull-ups. Remember, the enemy's got a truckload of party favors designed to take us out, one of them being functional amyloids. This realization has sparked collaborative studies with a broad range of scientific disciplines. While insights from these experiments has led to innovative treatment strategies, there are still knowledge gaps that necessitate further investigation. As daunting as this may seem, our adversary is persistent and will pursue any means to overcome our defenses. We must be just as resilient. Make no mistake, this is a fight for our very survival, and we can't afford to give any ground. And so we march on, till victory is won. Get some rest, soldier. I'll see you on the front line. Hoorah!